Hello, and welcome to the presentation on calcium imaging and neural activity model. So just as a quick overview, calcium imaging is an indirect measure of neural activity through calcium-dependent fluorescence sensors. It's a very versatile technique, so it can measure the activity of a singular neuron all the way to subcellular structures over long periods of time, making it good for longitudinal studies, as well as also ensuring precise visual localization. It's generally utilized to investigate synaptic plasticity and also explore neurological disorders or develop treatments following these calcium ions. The methodology was developed by researcher Cien, which caused a paradigm shift in the neuroscientific field in the 1980s. For the experimental protocol, the first step is to prepare transgenic mice with GCAMP, and this is a genetically encoded fluorescent indicator that makes neural activity visible as it binds to calcium ions for tracking. Then breed the mice and wait 8 to 12 weeks for a stable signal to make sure it's bright enough for imaging. Surgically begin cranial window implantation and put in a head plate for stabilization during the behavioral tasks, then allow the mice to recover for 1 to 2 weeks before imaging. From there, prepare 2-photon microscopy or wide field at the ideal excitation wavelength and record the behaviors via videos or task sensors with the head fixation bar. Then capture fluorescent time lapses and synchronize all of these imaging frames, and then complete motion correction and segment neurons into ROIs for processing and data analysis. The experiment that I chose to look at here, it was highlighting what role glial cells have in synaptic plasticity, steering TDCS in the mouse's cortex. Calcium imaging was used to visualize neurons and glial cells and capture changes over long periods of time while also maintaining high spatial resolution. It was kind of unique how calcium imaging enables the observation of large populations of cells over an extended cortical area. This image right here, it's basically showing the activity from the 2-photon image in the spontaneous trial and the trial using the TADCS. To differentiate between neurons and astrocytes, they used genetically encoded calcium indicators, which showed large synchronized calciums in the astrocytes. Which you can see here, the synchronization, whereas the neurons didn't have very little change, and there's no synchronization. And this leads us to the finding that the synchronization is a key mechanism for the transdirectional current stimulation-induced metaplasticity. Some limitations of calcium imaging. The first one is the indirect measures of spiking. Because it's not happening in real time, it cannot capture precise signaling. Rather, it is reporting the changes in intracellular concentration following the spikes. In addition, it also assumes non-negativity, so all of the negative deviations that are lower than the fluorescent baseline is removed, so anything that is an inhibitory signal is not counted. For brain motion sensitivity, this is why they put in head plates in the mice, because slight movement of heartbeat, breathing, commotion, or even just small ones in the head, it causes fluorescent signal recording issues. And then there's also somatic signal contamination. So any signals from other parts of the neuron that isn't just the soma, it causes overestimations in the data collection. These are all the sources I use. Thank you so much for listening.